GU Lab, and I will be presenting today on transcriptomic and genomic variation in large families of chemosensory receptors. I know a lot of you are saying, what does that mean? And that's kind of vague, and that's what we'll get into. So to start off, we study the vamronasal organ. So the vamronasal organ is located within the nasal cavity here, and inside the vamronasal organ, there's vamronasal sensory neurons, and the neurons express different receptors for chemosensory action. So what we see here on the apical side, they express the vamronasal type 1 receptors, and on this side, the vamronasal type 2 receptors. So these are proteins that are expressed that detect pheromones in the environment, and they project to the accessory olfactory bulb into two different locations, and every receptor projects to the exact same glomeruli on here on the accessory olfactory bulb. So when we look at the vamronasal receptors, we can see that the V1Rs right here and the V2Rs, the V1Rs have a smaller amino chain here, and this is because they detect more lipophilic small molecules, whereas the V2Rs have a larger N-terminus chain, and they detect larger peptides like major urinary proteins. So if a mouse smells another mouse's urine, it can detect that, the cues within it. So we also look at the amino acid conservation within the 7 transmembrane domain, because these are GPCRs. We see high amino acid conservation within this, showing that this is something that's evolutionarily conserved across the species and strains. So when we look at the chemosensory system, we know that it plays an important role in orchestrating sexual behaviors and evolution of animal species. What we don't know are the genetic mechanisms behind sexual dimorphic behavior and strain-specific behaviors. So that's what we'll look into. We studied by looking at tissue sample from the vamronasal organ. We did total RNA sequencing, and this is kind of our design. We had mice from 4 different strains, black 6, 129, SJL, and SWR. There was 4 of each replicate for each sex. We had 4 females and 4 males. So this allows us to get higher certainty in our differential expression calls. So we kind of know the background of the mice. They've diverged quite a bit over time. So when we look at the RNA-seq data, and we map this down onto the genome, we only have the black 6 genome that's annotated and ensemble. So using the black 6 genome and mapping the reads down, we can look at the uniquely mapped reads, the multi-mapped and unmapped. And so in this case, the uniquely mapped, they map to the same loci, whereas multi-mapped, they map to multiple loci, and we aren't confident in those calls, so we don't use them or the unmapped reads in our analysis. So we use a pretty stringent threshold on what we allow for uniquely mapped of 2 or 3, in this case 3 mismatches from the gene itself in the genome from the base truth. So in this case, the first thing we look at when we take this data set is we want to look at all the genes that were expressed, and we want to see the variation within that in a dimension that we can actually visualize, because we can't visualize as many dimensions as there are genes. So we project this data set down to a lower dimensional space using principal components analysis to try and maintain that variation or large-scale variation. So looking at this, we can see differences between the strains very clearly across all the principal components. So here we have principal component 1 by 2, and we can see separation between these strains very well. Whereas the strains SJL and SWR, we don't see as well separation, but in the other principal components, we see separation between all of them. The important thing to note here is that we don't see separation between the sexes very well as compared to the strains. So this is indicating that the overall genetic landscape is more towards sexual divergence or strain-based divergence than sexual-based divergence. So now looking at the sex differentially expressed genes. So for the few that are differentially expressed, we see that of all these are all linked to sex-specific alleles. So in this case, we don't actually see differential expression changes. We see sex allele-based expression changes. And when we try and look at specifically the expression of the alleles for gene silencing, we see that actually if you take into account the X and Y allele, the differential expression call kind of goes away. So we don't actually see differential expression between the sexes if you take into account both the X and Y chromosomes. So there is no differences between the sexes as far as we can tell. So then looking at the strain-based differences overall, we can see that there's this mosaic pattern where certain strains are expressing genes more than other strains. And sometimes it's specific to one strain and not found in others. Sometimes some of the strains share the exact same genes. So overall, there's a lot of genes to look at here. and this isn't really meaningful, so we'll dive into some of the subfamilies and look at those and kind of the meaning for us in our lab. So first of all, looking across the chromosomes, we can look for expression patterns. So here we look at the genes as black bars. The green dots indicate expression, the orange dots indicate differential expression, and the purple or pink indicates an area of hotspot where there's a lot of genes that are differentially expressed in that region. So there's some type of regulation event going on here that is causing a lot of genes to be expressed here in one strain versus the others. So if we go through here, we look at chromosome 5, we can see that chromosome 5 contains the VMN2R genes, or one of the clades of the VMN2R genes. Chromosome 6 contains a clade of the VMN1R genes. And chromosome 7 contains the olfactory receptor genes, which is another type of chemosensory receptor that we are interested in our lab. And then chromosome 17 has 2 different hotspots on it. It has the clade 8 VMNR2 genes, and it also has the MHC, or major histone compatibility complex genes that are differentially expressed. And these are to be as expected to see differences between strains. They have selective immune systems for antigen binding. So we're expecting to see a difference between them in terms of the MHC. So now looking at the gene ontology analysis, what we see is that the major histone complex is number 1. We see a very large differentiation between the strains in that regard. We also see differentiation in the pheromone binding activity and also peptide antigen binding and all these other ones that also covary with these two big groups. So pheromone receptor activity and MHC complex, we have peptide antigen binding and a bunch of different antigen binding ones here. So this basically is just saying that overall we see a lot of these different events. So now if we look at the expression or the genes that are shared between these different strains, what we see here is that a lot of them are shared. 1,200 of them are actually shared of the 1,500 total. So what that kind of tells us is that they all are expressing the genes. Some are just expressing them more than others. But what we're trying to look for here is saying, because we're using the black 6 genome, because we don't have other genomes for the other strains, we want to know if there's preferential treatment for the mapping of the RNA-seq data. So when we look at this, we see that there is a bit of preferential treatment to black 6, but we haven't yet well characterized this because we don't have annotations for the other genomes. So what we're looking for now is we're actually mapping the annotation for black 6 over to 129 to be able to see if that preferential treatment stays. And that'll allow us to be more confident in our differential expression calls. So here, looking more at specifically at the vamarinasal receptors, these chemosensory receptors, we're going to highlight specifically these 3 clades. And so what we see here is that inside these clades, these are vamarinasal receptors that share sequence homology up to like 90%, 99%. So what we see within our differential expression data is that actually there's differential expression in clade E here, and clade J here and here. And so we see differential expression for these 2 clades, but importantly for later on, we don't see differential expression for clade D. And then we also see the V2Rs have some differential expression calls themselves. So this is setting up something for later on that we'll talk about. But it's important to note that we see clade J and clade E have differential expression. So now looking overall at all of the V1Rs and V2Rs, we see this type of mosaic pattern where we have differential expression in a lot of different, these receptors, these chemosensory receptors, in certain strains and not in other ones. The important thing to note here is that we don't see a sex-based difference. We only see this strain-based difference. So this differential expression basically is showing that these strains are able to detect pheromone cues in the environment that another strain can't detect. So this is leading to some type of divergence between these species, or well, not species, subspecies, I guess. So in this case, we see that also in the vamarinasal type 2 receptors. Okay, and then one of the things we were also interested in is looking at regulation. So we looked at some of the long non-coding RNAs to see if maybe they are also differentially expressed in our data set. And especially because we have so many replicates, this type of data set is, when we see the differential expression calls, we can be more confident in it. So what we see here is that these long non-coding RNAs, we see a lot of them that are expressed in this black 6 versus the others. And again, we still have to look at the annotation on the other genomes once we get that mapped over to see if that's just due to preferential treatment or not. but looking specifically at some of these that in the mass action plot that are highly varying between the strains, we can see this MEAT in this gene model that are preferential to certain strains and not others. So we see high expression in 129 SJL and SWR, but we don't see expression in BLAC6. And here we see expression in 129 BLAC6, but not in SJL and SWR. So these types of patterns are important in looking for regulatory events or some type of molecule that has maybe possibly a mechanism that we can look into further. So when we look beyond just these two and look at the total and how maybe they vary with some of the vamorinasal receptors, so we take all of those long non-coding RNAs that we were looking at the expression plot before, and we look at them and their correlation to all the vamorinasal receptors. And we see a high positive and high negative correlation with the vamorinasal receptors, showing that there is reason to investigate this further and look for possible mechanisms of regulation of these vamorinasal receptors expression by these long non-coding RNAs. So this is one of the avenues that we're going to look down further in the future. So now looking deeper at the clades, I talked before about there's clade E and clade J, and then we didn't see differential expression in clade D. So the clades actually represent what type of pheromones can be detected in the environment. So these clade J receptors can detect estrus urinary secreting cells. The clade E is for female urinary secreting cells, and the clade D is for male urinary secreting cells. So before we saw that these two were expressed differentially between the strains, but we didn't detect D. And so we were asking the question is why? So looking into clade D, what they found is that there's actually a novel clade within D that's D-like clade that has sequence homology, high sequence homology within themselves, and they're very similar to the D-like locus. So looking at that locus, and we can see that this is the D-like locus that they found, that there's these genes right here have amino acid conservation of up to 100% or 98%. So what we see is that we have these motifs here across this 4 megabase region. So what we see is the motifs are repeating, where this orange and the two black and blue here are repeating down here in the same motif on both sides. And then we also have these red ones from this locus up here that repeat over and over again. So this is type of what you'd see in a duplication event across the genome, is you would see the motifs being conserved all across these reconstructions. And so when we look deeper into this across the strains, what we found is that low coverage is actually a problem in many of the strains. So when they did their reconstructions, they didn't have good coverage on the reconstruction of the D-like locus. So what this has a problem, other than BLAC6, this brings a problem up when we're trying to quantify the expressional changes for this D-like locus between the strains. We can't actually map down onto those strains accurately, so we're not getting good differential expression calls between them. So we want to do an accurate reconstruction of these strains so we can actually identify some of these differential expression or transcriptomic divergence, and also genomic divergence, so mutations or insertions and deletions that have happened in this locus between the strains, and how this associates maybe with sexual dimorphic behavior and the ability to detect male pheromones. So we looked at this reconstruction problem, and the first thing I looked at is maybe is there some new type of assemblers that can help us do this type of reconstruction. So because these sequences have high sequence homology up to like 98%, there's a problem with many of the assemblers is that they can't actually reconstruct something up to that level. So I looked at all the assemblers that were currently available, and I found this paper called Shannon on a new assembler that had been built recently, actually February 2016. And so I looked at one of their algorithms in specific, and I was interested in it as they were looking at isoform switching, and they wanted to be able to pull apart from one gene all the different isoforms that are being expressed in that gene. So they created an algorithm they called sparsus flow decomposition, which allows them to use an information theoretic model to deconvolute how the different isoforms are being expressed within one gene when they're doing their reconstruction. So what we did is we formulated this problem instead of isoforms, where they're sharing some exons and skipping others, where instead we're looking at these V1R D-like locus, where they share some sequence homology in certain regions and then diverge due to mutations, insertions, and deletions in others. So then looking at the initial assembly with Shannon, we got 8 reconstructions, which isn't a lot because we were looking more for like 90 genes. So in this case, with only 8 reconstructions, I could take and I can map them down and see where they blatted to the genome. And for this top one, for instance, we could see that as a partial V1R reconstruction. And this next one is like a flanking region to that. And we also got a reconstruction down here for a different gene that's in that same locus. So we weren't getting good reconstructions, and I was very interested in why we weren't getting good reconstructions. So I actually pulled apart the assembler, and I looked into how it was doing some of its choices in the assembler, like how it was assembling some of the reads compared to others. So what I found in doing this is that they actually were tossing out a lot of sequences that are very similar, and I turned that parameter off. So instead of tossing out similar sequences, which we're interested in, I allowed it to build contigs and allow for sequences to be very similar to each other without tossing them out. So in this case, we get more reconstructions. You can see the gaps because this is like a multiple sequence alignment. But the important thing to note is that looking vertically, you can see this shared homology here in these genes, and these are actually V1Rs. They map down onto V1Rs. And then this other gene family is the pseudogenes of the V1R D-like locus. And then we also have these genes that mapped to the same 4 megabase region that aren't a part of the V1R family. So the next thing that I looked at is, okay, so I turned off this parameter to allow everything essentially to be reconstructed without tossing out sequences that share some sequence homology. But what we got is instead, you look at maybe this sequence and the one above it, is that it's just a partial reconstruction of the one above it. It just stopped at some point. So I looked at, in de Bruijn graph assembly, you have to go each nucleotide at a time in your de Bruijn graph when you're assembling. So you only look for extensions that are 1 nucleotide extensions from where you're at. So if there's a mutation in that de Bruijn graph that you're building, it stops and it doesn't build any further. So what I wanted to understand is, can we actually look further and actually do a better assembly than this already? So I first started trying to merge these together. So if there was like a mutation, I could collapse down and just not care about it anymore. Well, what we ended up doing is creating a new way to calculate edit distance. So we converted ATC and G to these numeric representations. So let's we take 2 sequences like this and we have them overlapping. And even if they have a mutation here, we can represent the sequences as vectors of these numerics. So by converting A to 1, 0, 0, 0, we can have this sequence. And then we can pad the vectors so that they're the same size given the overlap. So in this case, it would have missing sequence here, so we would pad it with zeros. So now these vectors are the same size. So now we can do this as a matrix operation. We can actually multiply these 2 vectors by each other, and we can get out the resulting vector. So what this allows us to do is where the padding is, you're going to get all zeros. There's no matches between these. But where there's not padding, we can actually see that the sequences are the same here, and it's returned as being the same. But where we see this mutation event, it'd be right here, you see that 0 times 1 is 0, and 0 times 1 is 0, so you get all zeros here. So this gave us a way to calculate edit distance. So given this matrix, resultant matrix from this multiplication, or in this case a vector, we do it as a matrix multiplication in our algorithm. We can take the summation of this, in this case it's 10, and divide it by the overlap of these 2 sequences, which is 11 if you're the count between them. And we can get the percent similarity between the 2 sequences. So this is like a very fast way to do this type of edit distance-based calculation. The nice thing about this is doing it as a matrix operation instead of a string-based operation, is we can actually run this really fast on a CPU with dedicated libraries that are really quick. But we can also run this on a GPU even faster. So that's the nice thing about it. It's scalable when you can run it on a GPU. So the next thing is, let's say, okay, we want to do this overlap-based assembly, and we have a new way to calculate edit distance. So if we take a 100 base pair read, we say, hey, this is our seed read, and we want to extend onto it. So we look for possible extensions around it, and we say, hey, we're going to allow an overlap of 40 base pairs on either side. So then looking at all the reads that would stack up to that, and we can vary the similarity that we allow to match based off our similarity threshold. So we can get a heterogeneous mixture of reads. So these reads all map to here, but maybe they're slightly different from each other. So by allowing a different threshold, we get different mapping. So taking these reads, we can then cluster them to get homogeneous mixtures. In this case, what we're doing is we're separating out biological variation from technical noise. So as you know, in Illumina sequencers, there's some error still left over, even in current generation technology. So what we're able to do is actually cluster together and get read stacks, even though there's sequence errors within here, we can still see that there's the biological variation is being retained. So we're actually pulling that out. So I guess from here it's kind of difficult to see, but there's a TC mutation right here. Whereas in this stack, it's all Cs. And in this stack, that same mutation is all Ts. So we've actually separated biological variation from the technical noise. So we can still see that there's like Ns and mismatches of As and stuff in certain locations. And so what we're able to do is still assemble these reads even though they have errors in them. And we can do it very fast. So now taking these read stacks, like, well, what do we do with a read stack? We want gene reconstruction. We want to get full-length genes. So we can actually represent these as, the exact same kind of reads, and this is just like a better representation, so we don't have to look at these big ones. We can put the mutations back in there too. And we can start to form a graph out of these. So where one read window is unique and there's no other reads that stack up with it, we just put it into a node. And then if there's two reads that share the same window, we can merge them into the same node. So now you're wondering is like, what does the color represent here? Why is it all red? Well, I actually can give a similarity based off their overlap. So by looking up back in the matrix when I do this calculation is I can look up exactly the similarity of the two sequences here without redoing the calculation. And I can look up the similarity between the sequences here without doing the calculation again. So this makes it really fast, and we can actually show in a visual representation how good our assembly is or how much similarity is between the sequences. So by doing this, in this case with mutations, we can actually show there's a lower similarity between these two sequences that share the same window. And so we can fine-tune our assembler by doing that. And so we get, like in this case, two read stacks that are part of two different genes. So now as we start to build a better assembly graph, what we get is some things that look like this. So we have here what looks like an insertion event where one of the genes has some type of a biological insertion that you can see as this loop, whereas the other gene that was duplicated at some point that didn't have this insertion event just goes straight through and it doesn't have that insertion. And also the nodes represent the coverage, so the larger the node, the more reads stack up in that exact same position. So we can look at the similarity within the nodes. In this case, because it's so dense, we can't see between the nodes, but we'll see that in just a second here. And then we can actually fine-tune our parameters of the assembler to allow less or more similarity or divergence. So in this case, in this sequence, they share some sequence homology here maybe, and then branch off into two different branches. And in this branch here, they even branch off again. It's starting to form here. So now you're wondering is why isn't this graph bigger? Why don't we see a large graph? Because these genes are really long. So these are actually really long reconstructions, but we should see a large graph of all these things connected to each other. So the reason why we're not seeing that is only allow a 40 overlap. So they have to share 60 base pairs of their sequence together to be able to merge. So by using paired end reads in a larger overlap and that insert size distance, we can actually merge these things together. And that's the next step of this algorithm. So far, we've been working on it for about like a month or a month and a half. So as we start to develop it more and put paired end reads, we can actually form larger networks of these graphs and then show how the relationship between these different divergent events. So in this case, the next step of our algorithm is with any assembly algorithm, you're going to have over assembly where you're just matching things that shouldn't belong together. So in this case, like if we allow too low a threshold and what we allow for matching, what we see is that, like in this case, we have a low sequence similarity here, so like somewhere between 90 and 92 between these two reads. And they also have low coverage, so they're the smallest nodes in the stack. So there's not a lot of reads that stack up there. So it's not very trustworthy to keep that as a biological true signal. And so what we would do instead is we'd say, okay, this isn't real, we should cut this. So this is called pruning, so we can prune our graph into subgraphs. And we can pull apart the pseudogenes from the regular V1Rs, and then also pull apart the other genes that are in the same locus. And so then we can solve each of those independently. So then I talked about the Shannon assembler before and kind of what their algorithm does with isoform switching. So I'm going to walk you through like an example of that in terms of like a simple mathematics, I guess, example. So you'd have some genes that share, they have some unique regions on the left and right, and then they share some sequence in the middle. So this region is larger than our read length, so we can't actually differentiate where we should assign reads from here to this side. So instead, we can look at the flow between the regions. So in this case, we have a coverage of 6 on average going into this node, and we have a coverage of 9 here and a coverage of 12. So then going out of it, we can look at the flow on the other side, 5, 7, and 15. It's not very simple to solve this initially. You're like, what do I do here? But what you actually can do is merge together. Let's say we want to look at S1 and S2 and how they go through S3. We can merge these two together and say, hey, if it works, okay. If we merge S1 and S2 together to go to S6, we actually use up all the information contained within this branch or this part of the graph. So this is actually a very simple solution to this. And then for S0, what's left over, well, this is 12, these are 5 and 7, 5 plus 7 obviously equals 12. So then we can also make connections there and then merge them, and that's our reconstruction. So what this actually is doing is it's finding the simplest solution to this problem. So it's forming this as a graph, trying to say that this information that's here is real information, and we want to retain all of that in our assembly, and we want to make the simplest solution, the sparsest solution, kind of like an Occam's razor. So in this case, by parsing through the graph and calculating out all these different combinations, we can get out the true assemblies based off that coverage information. So what we've looked at today so far is that this transcriptomic data indicates that there's no differential expression between the sexes other than this allelic type expression on sex-linked chromosomes. So in this case, we also found that there's a lot of differential expression between the strains, and many of these are involved in chemosensory receptors transduction. So the other part that we also found is that the non-coding RNAs are possible regulators due to their high correlation and anti-correlation with these chemosensory receptors. So we're going to look further into them for regulatory type events or mechanisms. And then we found that the V1R D-like locus has high sequence homology. These motifs are kind of repeating due to these duplication events, but they have some slight difference. So using this type of overlap assembly approach, we can try and parse out this biological variation from the technical noise. And we can actually do this for any sequencing length or any type of sequencing platform. And another thing to note that I guess I forgot is that we can also take into account the Phred scores, so the quality scores of the sequences. We can put those as a vector and multiply them by the vectors of our sequences. So we can use the quality scores to actually make the sequences more divergent if there's less accuracy from the sequencer saying that this is the right call that I made, if this is an A, T, C, or G. And so that can allow us to assemble better. We haven't yet tested that out. Like, it works, but we don't know how well it works in terms of the total assembly. So once this assembly is finished, we'll try out and say, is it good to use the Phred score, or should we leave it out in our assembly? So this is kind of like what we've been doing in the ULab, and I would like to thank you for your time. This is the ULab. These are the people in it. Obviously, Ron Yu and Li Mei Ma. We have Rishabh Raj, which he helped me to make some of the mathematics for the vector operations, so we could get this assembly running really quickly. And then we're going to be moving it onto a GPU hopefully soon. And then I would like to thank Ariel and Hua specifically because they are part of this kind of team in trying to reconstruct these genes. So thank you. Yeah. So in this case, we're looking specifically at that 4 megabase region that I highlighted earlier. I know I kind of went fast for this. There's a lot to go through. Let's see. Animations are cool until you have to click backwards, I guess. Yeah, so this 4 megabase region is where we're looking at specifically, so they can actually pull that out with probes, and then we can look at specifically that region and not have to look at all the other types of receptors and their variations, which will make it a lot more complex. My hope is that we can look at further and more sequences and maybe see how those reconstruct inside of our algorithm. I didn't quite catch that. The DNA polymorphisms from the sequencing error, is that? I can't hear you. Sorry, I don't have that great of hearing to you. Yeah, so if it is some type of, in this case, we're looking at genomic data, so we're not going to see some type of editing, right, post-transcriptional modification. But what you're saying with polymorphism, that's what we would expect to see between the genes, right? We'd expect to see some type of polymorphisms between them. And so that's what we're trying to reconstruct by looking at specifically in one mouse that we had sequenced. We'd be able to reconstruct all those polymorphisms and say that, you know, this is something that is a real polymorphism and not sequencing error. So if I go to, let me get this back this way. So these, inside of this plot, we can see that there is, you know, sequencing error, like an N would be like where the sequencer can't make a distinct call on which base it would be. But we can also see like here, there's an A versus these T's. So in this case, if we were to sample from multiple organisms, right, and you were to see, you know, A's and T's, with multiple organisms with like a single nucleotide polymorphism, you wouldn't be able to tell the difference between the single nucleotide polymorphism in one organism versus the others. So it would be considered a sequencing error in this aspect. But if we take from one organism, we're going to reconstruct all of those. Yeah.
So I have a question, how your findings relate back to biology? So you have all these strain differences. Does this then affect your behavior? Does different strains mate with each other against?
Yeah. So we can't accurately quantify the expression of the different strains right now at that locus, and that locus is tied to the pheromone receptor activity. So once we actually can reconstruct for the different strains their D locus, then we can map the reads back onto there and accurately quantify and say, is there differential expression here? Because right now, when you try and map down, you're not going to get anything that lands, especially when we only have black 6 for the reconstruction, and we need the other ones, and we know that there's some type of sexual dimorphic or strain dimorphic behavior. That's not the right word, but we have some strain different behavior between these. So we know that there's some differences in that locus for how they detect male receptors or male pheromones.